I am Akio Ebihara from the GIFU University. So, I really happy to have these lectures at this institute. So, I am a biochemist. So I'm working on the protein science, especially in the protein structure. So, do you know the work of the Professor Hodgkins? Yes. And she's a pioneer of the X-ray crystallography. So without her, we couldn't use the X-ray crystallographies. So, do you know her? Yes, her. And she solved the structure of the structures of the ribosomes. Yes. So, one of the famous person from India. Do you know? The Ramakrishnan. Yeah. So, X-ray crystallography is very clear. So it shows us a protein structure. So for starters, I will show you what the protein structure is. Yes. And this is the protein structures. It is electron densities of the protein. A protein has no color. It just has the electron. See? Yes, you see? And we cannot understand what the molecules is. So by looking at this electron density, so we use a cartoon. And I think that you know the detail here. I think that you may see this type of the cartoon in your textbooks. And so X-ray crystallographers makes a protein crystal, and then we use an X-ray to determine the function, the structures. Based on these structures, so I use the structure analysis to evaluate its quality. So today I would like to show you one of the approaches. And the protein can be used as therapeutic purposes. And the protein is one of the biopharmaceuticals. And so one of the famous example is erythropoietin. The introduction of this recombinant protein, pure protein in our bodies, can help you the treatment of the patient with anemias. Another one is the antibody. Antibody can bind to the target protein with the specific manner. Antibody can recognize the specific site in the molecules. That's why we have immune systems. So with the antibodies, so the viral infection can contained. So the therapeutic antibodies can be directly to induce our immune systems and block the another immunogenic reactions and inhibit viral infections. So I would like to show you on the basic. And the protein is a chain of the amino acid sequence. It adopts the specific conformation according to its amino acid sequence. The functional groups of the protein are well defined in the three-dimensional spaces. And protein-based and pharmaceutical should retain and structural properties to exert its functions. So now I want to show you one example. And the protein is very flexible. So it's easily to degrade. So during your operation. So you may use the protein itself for your research. Other times, please imagine that protein is very flexible. And so when you mix the protein solution and vigorously, so it's easy to be denatured. I think I made this one by mixing the egg. You see that that merengue. You see, so it is and comes from the denaturation of the protein. So please handle your protein very carefully. Yes, this is one of my message, the practical message for use. And today I have two topics. And the first is the production of the recombinant protein. I monitor the structural properties to know the function. The other one is the proving of the protein structures using the antibody, site-specific antibodies. So I start this topic first. So as the chair introduced, I have worked on the one of the biological systems called renin-angiotensin systems. All of you have these systems to regulate your blood pressures. So antihypertensive drugs interact with these systems. This system is an enzymatic cascade to produce this peptide called angiotensin 2. And this peptide can increase blood pressure and regulate our electrolyte balances. Angiotensinogen is the precursor of this peptide. This protein here is cleaved by an enzyme called renin here. So I want to know how it works in our bodies. So I want to know the enzymatic reaction and from the point of the view of the substrate. A substrate is also important to understand its functions. Apart from the increasing the blood pressures, angiotensinogen have another function. It inhibits the making a new blood vessel. So recombinant, and the recombinant proteins and placed onto the thick membrane, new blood vessel, and the formation of the new blood vessel are inhibited. So the authors showed it has an inhibition. Yes, it inhibited angiogenesis. So to facilitate our research, we are going to make a recombinant protein of these proteins. So we have succeeded in making it in a mammalian cell. So we are going to make this protein using the E. coli. E. coli is different from the mammalian cell. So we anticipate that this protein making this protein may be very difficult in E. coli cell, but I want to produce this protein in E. coli. So to tackle this one, so we changed the several culture and conditions, but we couldn't obtain a good sample. They are all insoluble ones and very bad for our analysis. Now we change another and we use another strategy. So we use weaker promoters. So when we use weaker promoters, the soluble to insoluble ratio was increased to 1:1. So it means the protein holding is facilitated by the weaker promoters. So now we establish the production method, and we compare the recombinant proteins expressed in the E. coli with that expressed in the CHO cell and mammalian cell. And we use the various types of the analysis to know the function. It is something like the multidimensional approaches, and I use the several structure analysis to know its functions. Okay, first I measure the enzymatic activities. So using the enzyme renin. So I skip the detail, but I measure the rate of the reactions at the various concentration at the various concentrations, and we reported the data, and we know this constant to illustrate the enzymatic function. So yes, and we compare the E. coli with with that in the expressed in the CHO cell. So these values are essentially the same. So it means that a recombinant protein expressed in the E. coli works for a renin function. So we are very happy to have this result, and we want to know the structure itself. So this is a result of the circular dichroism and data. So structure and content are also similar. So it means that it is properly holded in the E. coli cell. And I use another analysis called and the DSF and differential scanning and fluorometries. So when you need the and so many so when you want to analyze your protein in a high throughput manner, and please use this analysis. I think this is very useful for you. And this method can monitor the thermal unfolding of the protein in the presence of the fluorescence dye. And this dye, so it's highly fluorescent in the non-polar environment, which is similar to the inside of the protein. So, we obtain this type of the chart. So it means, so at the low temperatures, the protein is holded. No dye is entered in the protein. As the temperature increases, the proteins may denatured because it is fluctuated, and then to dye is incorporated, and we have the higher intensities from the dye. So you can analyze your protein in a 96 well. So it is very fast. It is very fast, and this is the data. And based on data, so we estimate the protein and stabilities. So and based on these data, thermal stability are essentially the same. So this is final data, summary. So our protein expressed in the E. coli is comparable with that expressed in the CHO cell. And now we make the protein crystal and to determine its structures. Yes, and when you have a protein, so you need to store it. So you need to store it. That freezing is one of the options. But you know that the freezing. So when you freeze the protein, you need a freezer. You need a freezer. So it is very useful for us to store the protein without a freezer. So we try to examine these possibilities inspired by these research papers. The authors reported the Puran-based encapsulation of the enzyme and its substrate. So, the Puran is not sugar. So, the authors makes a tablet, and the tablet is water soluble. So, you can use the proteins by the dissolving the tablet in the water. So that the data says the enzyme and its substrate are stored in a solution at room temperatures and becomes inactive within weeks. On the other hand, enzyme and substrate are stored in the tablet and remain active within a few months. So I want to use this method for the longer storage and without refrigerators. So the next one is the and probing the and protein structures using the antibody. So our next target is the protein receptors and called PRO. And this receptor is a single span membrane protein. So the renin, renin and prorenin can bind to the receptors to lead the angiotensin generation and the intracellular signaling. Interestingly, this receptor function as a cofactors of the Wnt beta catenin signal. So these receptors can bind to the component of the receptor and the complex. This complex is involved in the development and the cancer. And the recent report indicate that this receptors is crucial for the Wnt signaling dependent genesis of the pancreatic cancers. So when the pancreatic the cancer cell was treated with the scrambled RNA, so we see the significant tumor here. On the other hand, when the cell was treated with the siRNA for the PRO, so the cancer size is very small. So it works for the genesis of the pancreatic cancer. So this type of the cancer is very hard to find. So we are going to we want to know how the structure is like to make the the drug. So now at this present, no 3D structures has been determined. So but we have the model produced by the computers. Yes, it's published in that the 2015. So we are going to make so we want to determine the protein structures, and we are making the recombinant protein now. But first we evaluate these the structures using the antibody. We prepare the anti-peptide antibody against the sequence specific for the PRO and carried out the binding assays with a panel of the antibody. So yes, so this is the protein structures. Some sequences are outside, some sequences are inside. So when we use the antibody, we may know which side is outside or which side is inside. So that's why I use several antibody against the specific site of the PRO. And our question is the epitope is the outside or inside? So we make the 9 kind of the antibody and specific to the PRO. Yes, a 9 antibody. And we produce the antibody and the protein and the PRO, and we examine and we use the Western blot and the binding assays and using the antibody. So today I show you a summary of the binding assay. And we use this methodology biolayer interferometry. So, and this method measures the interference of the white light from the two phases. So I skip the detail. So, but when analyte bind to the biosensor, and binding signal can detected in real times. So we monitor this type of the data to know the binding. And we made the premix solution. We mix and PRO and with the respective antibody prior to the binding assay, and we use the protein A sensor to detect and to capture the antibody, and we have this type of the chart. On the other hand, we use the antibody alone for the reference. So and this difference is attributed to the antibody binding to the PRO, and we obtain this type of the data, and this 1, 2, 3, 4, 5 site, and gives us the bigger values, the bigger binding signal. And we evaluate the protein structures using our weight data. So and the 5 site with us high signal are located in the outside. So we say, and we can say that the model is not acceptable at this present. So now, we know that, and we have several. We found several antibody, so that can bind to the PRO. So we hope that yes, and we are going to make the blocking antibody for the pancreatic cancer. If it works well, so we can make the therapeutic antibodies for the pancreatic cancer. So the pure science could be useful in our, the medical science. So this is the a suggestion of my works. So and briefly, the protein is prone to aggregate. So when you use your the protein, so please take care of it very carefully. So don't make a bubble. Don't make a bubble. And the antibody could be useful to make to know the protein structures, and it could be useful to. It is the candidate of the blocking antibody and the neutralization antibodies. So okay, I skipped it. And okay. And I'm also interested in the diagnosis of our body. So and we are going to make this type of the kit. Do you know it? And this kit is very useful to know that pregnancy. So when when you add the urine and you can know you are pregnant or not. So I want to develop this type of assay against our target and prorenin. And prorenin is the early markers of the complication of the diabetes. When you have a severe diabetes and you have the diabetes, the diseases for the longer times, you may be blind. So it is very, it is very tough for us to lead the lives. But prorenin is the early markers. It means that, yes, that the medical and research says the prorenin level is increased 5 years before the complication. So we may detect when the complication occurs. So I want to make this type of the kit to improve the quality of life and QOL. Okay. And yes, so okay, and this is acknowledgement. So my student works so hard to do this research. And okay, I have one thing. So this is the data that relating to me. Do you know? And what do you think? Yeah. So year, yes. And what is it? Oh, no publication. No. Age, no. So.
Is it your break in career or something?
No. This is this data strongly related to my body.
Oh, body weight.
Body weight, yes. And body weight. You see? And at 55,
You are getting fitter.
90. So I think this is the data when I was the junior high school student. Actually, so I am the judo player. And do you know the judo? Yes, judo. So at the times, I eat and eat. So I need to take an entrance exam for the at the university. So I stopped the judo, but I couldn't stop the tendencies. So I have sharp peaks here. And then I realized that, my features is so dim. So I need to reduce my body weight, of course. And then I reduce the food amount. So I can reduce my body weight about 10 kilogram one year. It is very so hard for me. And then it keeps on the plateau. And then something like that. But please look at this one. Very it slowly decreases why I change the food. Okay, skips. This is this is example of my lunch. My lunch.
This is the fermented rice.
No, this is
Oh, good point. Good point. Yeah, good point. Yeah. So, and this and they are and it is and brown rice, no polishment. Yeah, yeah. So, yeah, yes. So, I eat the brown rice. So, and this is very tasty, you know. And this is radish, Japanese radish. It is a kind of the sabji, yeah, sabji, yeah. and this is the a pumpkin, and this is a potatoes, and this is and the sweet potatoes. So just the steamed. So I don't use an oil. So I think this and brown this rice is very useful to reduce the body weight. Yesterday, yes. So you are working at the medicinal herbs or the food and specific in this field. So I realize that you have big treasures. So how to utilize it is very important for you and for me. Yeah. So I am the one of the examples. So I have visited the market at the Guwahati and before. So I met this guy and he showed me around. Yes. So I was very astonished to see so many kinds of the fruits and the 20-kilometer bottle. Yeah, so I was very astonished. So, and relating to these things, and we have symposiums at an IITGs next year. So the topics is future utilization of the bioresource in the northern part of the India. It is held in the February, so 1 to 4th. So IITG and the GIFU University and jointly organize these symposiums. So if you are interested in this the symposiums, and please come and join and have a discussion. So the abbreviation is IJBS 70. So please check it. Thank you very much for your kind attention.